You'll get a quiet report.
Okay, so let's get started. We spoke last time, we finished up on a brief discussion of sequencing just as an example of the way in which DNA polymerase works. And we saw the use of a template, a primer, and then the extension. Now we're going to look at the enzymatic apparatus that does this. Specifically, we're going to look at DNA polymerase. And Arthur Kornberg, we now know that there are a number of different types of DNA polymerases, but Arthur Kornberg was the first one to isolate a DNA polymerase after the publication of the proposed structure of DNA. So the structure of DNA was published in 1953. Within 5 years, he had isolated this enzyme, DNA polymerase, that could replicate the DNA. The very next year, he got the Nobel Prize. This was big news indeed. So these are the properties of the DNA polymerase enzyme, as described by Arthur Kornberg. He started with E. coli, an extract from E. coli, and from that he isolated a protein that, in his experiments, could convert soluble nucleotide precursors. So he would put ATP, CTP, GTP, TTP into his extract, and those soluble nucleotide precursors could be converted into a high molecular weight product. So that was evidence of the polymerizing ability of this enzyme. But he didn't get the Nobel Prize because he found an enzyme that could polymerize nucleotides. He got the Nobel Prize because this enzyme needed a template. Because it needed a primer and it needed all 4 nucleotide triphosphates. These are important points because its need for a template and its need for all 4 nucleotide triphosphates when he's using natural DNA indicates that this enzyme is designed to replicate the template and can only do so if all complementary nucleotides are there. That's the interpretation of this. And he also found that this goes 5' to 3'. So needing a template was a conceptual requirement. Needing all 4 nucleotide triphosphates was a conceptual requirement and really the clincher to argue that this was an enzyme that is designed to replicate DNA. Needing the primer and going 5' to 3' are mechanistic issues that were to pose conceptual problems in replication, as we'll see very shortly. This going 5' to 3' issue is something in particular that's going to cause a problem when we start to think about replication after a bit. Turns out that all such enzymes that have ever been looked at go 5' to 3'. No enzyme has been found that goes 3' to 5' in synthesizing either DNA or RNA. So let's look at the mechanism here. Here is the template strand. We saw this in the sequencing reaction. Here is the growing strand. And the part of the strand that pre-exists is, by definition, the primer. And it grows by having nucleotides added to its 3' end. So this is a 3' hydroxyl, which has been ionized and ready for attack, as we'll see in just a moment. But here is the incoming nucleotide. So you have an A paired with the T on the template strand. That was the conceptual requirement that is enzyme satisfied. And so you can see the A, the ribose, and the triphosphate here. And it's lying diagrammatically in this active site. It turns out that all of these types of polymerases have very, very similar active sites, which have two bound magnesiums in them that are immobilized in the active site by aspartate residues. Two of them are shown here, but there are other residues that bind those magnesiums in the active site. And these magnesiums are very important for carrying out this reaction. So diagrammatically, this is shown here. This magnesium is stabilizing the ionized 3' OH so that this can engage in nucleophilic attack on this phosphate. This phosphate is held in position by both of these magnesiums. And you know from your studies of enzyme mechanism that immobilization of the substrate is an extremely important component of enzyme mechanism, because only when the substrate is perfectly still, or almost perfectly still, can it be oriented and subject to attack, as shown here. So this is stabilizing this nucleophile, which attacks this immobilized phosphate. Contributing to immobilization of this phosphate is immobilization of the entire triphosphate with this magnesium, and this other magnesium here. So both magnesiums are participating in that. Attack here then leads to cleavage here, and this pyrophosphate then leaves the enzyme active site. And this A will have been added to this growing chain. So everything that we've talked about here is what's happening in this diagram down here, right at this spot. Here's the incoming A that we were just talking about up here. It sits in here, all of these things take place, and it gets added to the chain as shown here. But then something that's not shown here has to take place, and that's translocation. Basically, you have to take this double-stranded DNA and pull it to the left a notch, so that you free up this insertion site. So this is the insertion site. The incoming, in this case ATP, sits in the insertion site. Insertion takes place. In other words, it gets added to the growing chain. But then the whole duplex has to be yanked over one notch in order to free up the insertion site for the next nucleotide that comes in. So the chemistry takes place, and then a translocation step needs to take place. And we'll study translocation actually in more detail when we look at RNA polymerase, but just realize that that's an important step that actually must take place. Okay, so this is what happens. This is the nitty-gritty of replication, what happens at the active site of the enzyme. But if we look now, if we take a more global perspective, we can ask, what is the overall molecular approach to replication? We started here with what happens at the active site. what must happen to free up the active site again. But now, more globally, when you take a DNA molecule, such as this one, for example, if you take this DNA molecule and you want to replicate it, how does it work globally? Do you just take this duplex and make another one? So the red one is the new one and the blue one is the old one? Or do you take this duplex and just replicate it in scatter patches throughout so that you end up with two duplexes in which the new and the old DNA is sort of mixed up together? Or is it orderly like the replication of the duplex, except that it occurs what's called semi-conservatively, where each one of these strands replicates itself so that you get two new duplexes, in which case each one has a completely new strand, which is a copy of the old strand. So these are three ways in which you can imagine replication taking place. And it was a conundrum at this time. How does this happen? So Messelson and Stahl did an experiment. An experiment that was very important and widely admired. And the rationale of the experiment was this. Take and grow E. coli in N15 medium. Now N14 is the normal isotope of nitrogen, so N15 is heavier, denser. So this is heavy nitrogen. And so they grew E. coli for many generations so that all the DNA in the E. coli was of the dense variety. Then they switched to a medium containing the normal N14 for one generation of E. coli in order to see what happens to the DNA when it gets replicated once. If it's conservative replication, then they should end up with one duplex, which is the original dense DNA, and one which is now the light DNA. If, on the other hand, it's this dispersive mechanism where both strands are replicated intermittently so that you end up with the new and the old mixed, then you would expect two duplexes generated from this original dense duplex, each of which is somewhat less dense. Not completely light like this, but not completely dense either, midway in between. Their experiment, by looking for whether the DNA is dense or light or in between, could distinguish this mode of replication from this mode of replication. But it could not distinguish between this mode and this mode after just one generation, because the semi-conservative mechanism would copy this strand completely and this strand completely, still to give two DNA duplexes of intermediate density just like this. But when they go for one more generation, then they can tell the difference. Because if it's semi-conservative, each one of these strands will now duplicate itself to give two more duplexes of intermediate density and then two duplexes of the light density. Whereas if it's dispersive, it would be four duplexes still of intermediate density. So by switching to normal medium for one generation, they could see whether replication was conservative or either of these two, semi-conservative or dispersive. And if it turns out to be this, one more generation will tell which one of these it is. So that was the rationale of the experiment. The experimental method for this experiment then required a way to determine the density of each of the DNA types of DNA molecules. And the way that this is done is by so-called isopycnic centrifugation or equilibrium density centrifugation. The rationale behind equilibrium density centrifugation is that you take a tube with a gradient, usually of cesium chloride, maybe cesium sulfate for certain types of experiments. This was cesium chloride. Cesium is a very heavy atom, or in the case of cesium chloride, an ion. So they can make a gradient of cesium chloride here, which is very dense at the bottom and less dense up at the top. And if the range of density in this gradient extends from being less dense than a DNA molecule at the top to more dense than a DNA molecule at the bottom, and then if DNA is mixed in with this or applied to or in some way introduced into this gradient, and if you centrifuge to generate a very, very high force field for a long period of time, the DNA molecules will eventually migrate to an isodense region of the gradient. So if it's a very dense DNA molecule, like one of these, its density will match the density of the gradient near the bottom of the tube. And after centrifuging for a long time, all of the DNA will migrate to this part of the tube where it has the same density as the cesium chloride. Any DNA that is below this spot will rise because it's in cesium chloride that's more dense than it is itself. Any DNA up here will sink in the centrifugation field because it will be more dense than the cesium chloride up here. So the very dense DNA would form a band in this tube down near the bottom. Half-dense DNA would form a band in the middle. And light DNA would form a band up near the top. So in this way, the DNA that is dense, light, or in between can be separated from each other. The way this experiment was done, the position of the DNA in the tube was determined by a beam of UV light shining through the tube. The centrifuge is set up to allow this so that the UV light scans the tube from bottom to top, and DNA absorbs UV light at around 260 nanometers. So a band at the bottom would be detected as a band of absorption, as shown here. So this is the bottom of the tube. This is the top of the tube. And light DNA will be closer to the top, and dense DNA will be closer to the bottom. And what they found was that at the beginning of the experiment, as expected, there was one band of DNA at this dense position in the gradient. But then after one generation, there was still one band of DNA, but at a lighter position. It was now at a higher position in the tube. And so interpreting this, you have to ask, what kind of replication would give you still a single species of DNA, but lighter? Well, it couldn't be this, because this predicts two species of DNA. And so after one generation, the possibilities have now been narrowed down to semi-conservative or dispersive. After two generations, 1.9, After 2 generations, you can see that there are 2 bands. One at this intermediate density position, and one at a lighter position. You can see that rules out dispersive, because if dispersive replicated, it would continue to be dispersive, and you would get more white DNA, but it would still be one kind of DNA. It would be lighter, but it would just be one species. However, if it's semi-conservative, you would still have 2 discrete species of DNA. One completely light and one half light. And that's exactly what's found here after 2 generations. So they concluded this is semi-conservative replication. And then in case anybody thinks they might have made an accident in locating the exact position in the tube of these various species, they took the original DNA and they took these 2 species of DNA and they mixed them together. And they got 3 bands. So this demonstrated that there was no misunderstanding of their experiment. They are looking at 3 species of DNA, a heavy, an intermediate, and a light. So that's the Michelson and Stahl experiment that John Cairns, who we'll see in just a moment, in fact now, thought was the most beautiful experiment in all of biology. So Cairns was also interested in replication, but he was an expert in a different sort of technique, namely electron microscopy of DNA. In this case, of labeled DNA. The Michelson-Stahl experiment was done in 1958, and it showed that the replication of DNA was a very orderly process. This appears to be, this model would suggest some sort of disorder, but this finding of theirs showed that there's an underlying, very orderly process at work here. But Cairns wanted to see it. And so he did this experiment. In this experiment, he grew E. coli for a couple of generations in the presence of tritiated thymidine. So tritium is a radioactive isotope of hydrogen. And in this way, he made the chromosome radioactive. It was then spread and placed over a photographic emulsion of the type used for printing photographs in those days. And the photographic emulsion would produce silver grains in response to being energized by decaying tritiums in the radioactive DNA. So that you can see now where DNA molecules lie over this photographic emulsion. And of course, he looked at hundreds and hundreds and hundreds of E. coli molecules, having to find ones that were nicely spread out, not bunched up, ones that were in the middle of replication, and ones where he could interpret what the patterns meant in the middle of replication. And you can see up here his interpretation of this pattern. He is saying that this original blue circle here, which is the original unreplicated strand of the chromosome, corresponds to this, all the way around here. But replication has taken place over part of the chromosome, giving rise to this extra loop. And that extra loop extending over the top is this one in his interpretation. So the original chromosome was like this, but part of it has been replicated, and so you have this extra loop that extends up like that. Very orderly indeed. And so he was very pleased to see that. But it raised a question. There are two spots here where this loop joins. Does that mean that replication is occurring from one of these and, say, moving around the chromosome this way in a unidirectional way? So let's say this is the origin of replication, and replication has proceeded this way. Is that what this means? Or is this bidirectional replication? Is there a replication fork here and one here, and they're both moving this way and will eventually meet at this position? In other words, is this replication loop bidirectional? Replication having started here and then moving out. So in order to answer this question, this experiment was done. Label again with tritium at a low specific activity to label the replicating regions of DNA, just so that you can see where the replicating regions are. Then label very briefly with tritiated thymidine again, but at very high specific activity, so that you can determine the exact locations of the replication forks. And using a similar technique to this is the result that was obtained. Remember that DNA was labeled at a low specific activity to label the entire region undergoing replication. And so you can see here is DNA that's been replicated. It's labeled at a low level in between here, but you can see where the strands are. But then the brief high specific activity labeling has labeled this and this, the top and the bottom. In other words, this confirms this particular model of replication fork activity. The replication forks are moving bidirectionally from a common origin. This is the common origin, and the replication forks have moved this way and this way. And moreover, both strands are being replicated as the forks move. So it's bidirectional, which is one issue, and both strands, which is quite a different issue. And this presented a major conceptual problem, one that I mentioned way back at the beginning. When I pointed out that these polymerases all go 5' to 3'. So if the polymerases are all going 5' to 3', they should be able to replicate only one of these. Because one of these strands, the template, is 5' to 3', and the other template is 3' to 5'. And so how, if the enzymes can only go 5' to 3', how can they replicate both strands simultaneously? One really expected to see one strand replicated on this side and the other strand replicated on the other side. So this is the conundrum. This is the direction of replication fork movement. This template strand is 3' to 5', so this we can understand. The polymerase is moving 5' to 3', and it keeps moving, and the replication fork keeps moving, and replication continues. But how does replication occur here? So one model, which introduced a little bit of messiness into how all of this works, but that would explain the picture that we were just looking at, is the idea that while this strand is replicated continuously by a polymerase that sits here and just keeps on going, 5' to 3', this strand, called the lagging strand, would be replicated in little bursts of activity this way. So it's called the lagging strand because replication has to go this direction, which means that you have to expose a region of single-stranded DNA that will allow the polymerase to get on here and move that way. And while this is moving that way, this one continues this way and exposes more single-stranded DNA, and then another polymerase gets on and goes that way, and eventually these little pieces are all joined together. And somebody named Okazaki tested this idea using this experimental approach. He labeled E. coli DNA for the indicated times and the results over here. So for various lengths of time, the E. coli DNA was labeled. Then the DNA was isolated. And the idea was that these were growing cells, and so DNA would be isolated in the midst of undergoing replication. And then he wanted to ask, could he find these little pieces? Could he catch them before they're joined together? And perhaps even do a time course and watch them getting hooked up into high molecular weight DNA. So is he going to be able to find some small pieces that then get joined together into big pieces? And so this was a time course experiment. In order to distinguish small DNA from large DNA, he took the DNA that he isolated and centrifuged it on a sucrose gradient for a number of hours. A sucrose gradient that was at a very high pH so that the DNA would be denatured. He wanted to look at denatured DNA because he wanted to pick up these small pieces. And he wouldn't be able to pick them up as small pieces if they were still in duplex form with the high molecular weight original template DNA strand. So he had the centrifuge in sucrose under denaturing conditions. And then the idea was here is a tube. This is a different kind of gradient from the cesium chloride that we were just talking about. This is a rate centrifugation rate experiment. This tube has a solution of sucrose in it. That's a little bit more dense at the bottom than at the top, but that's just to keep it from getting mixed during the experiment. This sucrose is everywhere less dense than DNA. In isopycnic centrifugation, remember, the top of the tube was less dense than DNA, and the solution at the bottom was more dense than DNA. So the DNA would end up, after centrifuging for a very long period of time, it would end up at an equilibrium position somewhere in the tube. This is a rate experiment. So the sample from the experiment over there, after isolating the DNA and denaturing it, is placed on the top of this sucrose gradient. This is the sample of DNA from Okazaki's experiment, just extracted from the E. coli cell. And then this tube is centrifuged. It actually spins out sideways. Those of you who've done this are familiar with that. But we're drawing it vertically in the direction of the G-force. And the DNA here is being propelled into the gradient of g-forces. And if the DNA is small, tiny little pieces of DNA, it moves slowly. This is sort of intuitive. And if the DNA is big, it moves quickly through the gradient. And so big DNA ends up at the bottom, small DNA is moving downward, and it'll eventually reach the bottom. But if you centrifuge for only 10 hours or so, as they did over there, then you'll catch it in a situation where the big DNA has moved down to here but hasn't reached the bottom yet, and the small DNA is just starting to move in. And in this way, you can see how much small and how much big DNA you have in the experiment. And if these experiments are done by punching a hole in the bottom of the tube and collecting drops, or else putting a device at the top of the tube and sucking it out one drop at a time. Here, this was taken off the top of the tube. So this is distance from the top. And each one of these data points corresponds to the amount of DNA found by counting the radioactivity in each drop that's taken off from the top of the tube. So it turns out that if labeling is carried out for a short period of time, 5 seconds or 10 seconds or even 30 seconds, you get most of the radioactivity at 5 or 10 seconds. Almost, there's not much radioactivity because the period of labeling is so short, but what there is almost all right here close to the top of the tube. As you label longer, you get more DNA at the top of the tube, but you begin to find DNA that has gone farther into the tube as well. As you label for still longer periods of time, you begin to see that there's still some DNA at the top of the tube that's labeled, but now most of the DNA is at the middle of the tube. And if you label, this is at a different scale here. You notice at this scale, this is 10, 1, this is 10. Now, here is 60 seconds on this new scale. Most of the DNA is here. But at 150 or at 600 seconds, you find now that almost all of the DNA is at the middle of the tube. Meaning that at early times, he is indeed seeing these short pieces of DNA being produced, but that as time goes on, after 10 minutes, virtually all of this low molecular weight DNA has been joined together into high molecular weight DNA. To this day, therefore, these little fragments are called Okazaki fragments, after Okazaki. So we're going to go back after a bit, next time, and we're going to look at how the polymerase accomplishes all of this. It's a very tightly regulated. It looks kind of messy when you think of it now. It's conceptually messy to have to replicate this as one continuous strand and replicate this as little pieces. But it turns out that that's the only way it can be done for polymerases that only polymerize in the 5' to 3' direction. So it's very tightly controlled. We'll see that. But let's look a little bit more at the mechanism of the polymerase as it adds nucleotides to the growing chain, and how it selects the nucleotides to add on, and how it remains so specific. How does it choose so carefully and make so few mistakes? Now, as I mentioned before, when I was talking about RNA structure, remember, compared to DNA structure, I pointed out that in the double helix, you have virtually absolutely correct base pairing in the double helical DNA and genomic DNA, all up and down the helix. It's all correct. But that's not because the helix is unable to accept unusual base pairs. A double helix can tolerate aberrations in its structure and little bulges here and there. We know that when we look at RNA structure, because RNA has such a vast array of structures possible, and all kinds of helical arrangements are possible. So the reason the DNA double helix is so absolutely correct in its base pairing is not because the helix is fussy, it's because the polymerase is fussy. And it was thought for a long time, it was assumed for a long time, that the polymerase supported its fussiness by insisting on base pairing being correct. But it turns out that although base pairing is important for double helical stability, remember the stacking creates the hydrophobic interior to the helix, and that hydrogen bonding then becomes an important stabilizing force running up and down the middle of the helix. Although hydrogen bonding is important for that, it turns out that for many polymerases, hydrogen bonding is not important at all. What, according to this slide, is important for this DNA polymerase in choosing the right base pair? It's an idea we've actually talked about several times already.
Shape of the active site?
Yes, the shape of the active site. And how is the shape of the active site important?
The nucleotide must fit the...
Absolutely. The nucleotide has to fit in the active site. And only if it fits exactly right is it accepted. And that discrimination is so accurate that the polymerase can distinguish almost absolutely correct from incorrect base pairs. However, this was an idea that met a lot of resistance for a number of years when it was first introduced, but now it's pretty much accepted. So this illustration here is very diagrammatic, of course, and so you have an active site here into which a CG base pair fits. Now we've seen before in RNA that you can have GA and you can have CA and various kinds of other pairings besides the Wasson-Crick pairing, but a CA base pair would not fit into the active site that's diagrammatically illustrated here, because it's joined to the sugar phosphate backbone and it's tilted in a different way. And so, as illustrated here, this purine ring just could not fit in because the active site boundary is here. And similarly, TG wouldn't fit in because the T, in order to hydrogen bond with the G, in order to fit in there, would be pushing out the top of the active site as drawn. Similarly, you have a TA base pair here fitting in the same active site. Remember, we looked at how the base pairs are the same shape, regardless of whether it's AT or GC, and regardless of whether it's this way or this way. And so this illustrates the idea of shape. And so the question is shape in fact sufficient to discriminate? And in order to ask whether shape is sufficient to discriminate, what would you have to do? You'd have to make a base that has the right shape, but it can't hydrogen bond. Make a base that has the right shape, but that cannot hydrogen bond, and ask, will it be incorporated in the way you predict? This is a cool experiment, both figuratively and literally. He is the one who generated this controversy by suggesting this concept, and he's devoted a huge amount of effort to establishing that this concept is correct. So this experiment, which was done a number of years ago, so you can see the controversy ran for a number of years, began with this molecule, difluorotoluene, attached to a sugar. So here are the two fluoride atoms replacing these oxygens. So these cannot hydrogen bond like an oxygen does. So this is a non-hydrogen bonding molecule. Doesn't have a nitrogen here, so this is not a hydrogen bond donor. But it has the same shape as a thymine. And the experiment was done as follows. First of all, he managed to incorporate this into a double helix, and he showed, consistent with the idea that it cannot hydrogen bond, that a double helix containing this is quite unstable relative to a double helix with a thymine. So in other words, the double helix was still able to stack, but it could not hydrogen bond at every AT base pair, and so its stability was reduced, corresponding to the loss of those hydrogen bonds. So the experiment that he did was to take a template strand that was one base longer than a primer strand. The primer strand was 23 nucleotides long, this was 24 nucleotides long. At the end of this template strand was a nucleotide, and in the experiment he would add various nucleotides to the reaction mixture in the presence of DNA polymerase, and ask whether the DNA polymerase would add that nucleotide to this primer end, given the X, whatever that nucleotide is in the template strand. And the results are shown down here. Different bases were placed at the X position in the template. And then for each one of those template positions, one of 5 nucleotide triphosphates was added. A, C, G, T, and F was the 5th one. And the question was, which ones will get added